Hi, Bartonella buddies. I just wanted to let you know that I have timestamps so you can skip ahead to each section, but our interview was more of a conversation, so we do circle back around to topics throughout. And if you found any information in this interview helpful, please consider sharing it in relevant Facebook groups if allowed, and on Reddit and other forms of social media. Thanks, Bartonella buddies. Hi, Bartonella buddies. I'm here with Dr. Marna Ericson, and instead of me reading a long, boring bio, I am going to let her introduce herself and tell you how she got interested in studying Bartonella and Bartonella in the skin and all that jazz.
Hi, Jake. Hi, Bartonella buddies. It's an honor to be chatting with you all today. My name is Marna Ericson. I live in Minnesota. I did say that just with a Minnesota accent, I think. And anyway, I'm a PhD and I have been working on Bartonella now, let's say 10, 15 years now. I know how long I've been working on it because that is when my son, who was 14 at the time, came down with Bartonella. And I have been in the Department of Dermatology, the University of Minnesota, doing advanced imaging on skin. And particularly, I've been using confocal microscopy and variations of that to look at the skin and other organs as well. And what we can do with that is we can do 3D imaging of specific organs, organ systems, microbes, whatever we are interested in. So my son got ill somehow. We're not sure why, as I know is a common thing. And it took us 50 doctors and through a chance meeting with Dr. Breichford, he analyzed my son's blood to find that indeed was infected with Bartonella Hensley. We think what was probably cholerae and Vinsonii. Well, then that started the next part of the journey once we had figured it out. And then, of course, I will always say, well, can I have a biopsy? And people who know me will know that's what I'll ask for. So we took biopsies. My colleagues in dermatology took biopsies. He had no visible lesions at the time. So we took them from his calf. And because he was a 15-year-old boy at the time, a scar was cool. So then we have more of those biopsies from that same area. And the reason I bring it up is because what I have found with the work I've done is you don't need to biopsy a lymph node, a lesion, Bartonella tract, and we'll talk about that in a bit. You can pretty much biopsy anywhere and you will find it. It is more abundant in some of the lesional tissue than in the non-lesional tissue, but nonetheless, it is still present. And I think that is an important point to note when we're talking about the distribution of these different microbes. And I am now working with, still at the University of Minnesota as a fellow at the Hormel Research Institute. And I am a fellow with Think Lead Innovate Foundation out in DC. And I am working now with Bob Mosiani out in Washington, DC. Mm-hmm. And we're putting together the research and the science and the clinical together.
Love that. About it being found all over the skin. We were talking about this before we hit record, but we could talk about it again. We talked about how it makes sense that it's found all throughout the skin if you think about the vector biology of Bartonella. And so it would be found all over the skin in order to be taken up from one host and spread to a new mammalian host. Correct?
Yeah, well, kind of, yeah. So,
okay.
I mean, most people, not everyone, will get Bartonella through a bite or a scratch in the skin. The skin has layers, that outer thin epidermis that's constantly growing and sloughing off, and below that is the dermis, the thicker part. That's where most of that's where the blood vessels are and most of the nerves, and below that you've got fat and muscle. So what we have found when we do what's called a full thickness punch biopsy, and it's like if you had like this pen, and you went in and you kind of just took a little punch out of it. So that allows us to look at all the layers of the skin. And we have found, and I'll say, so my first foray into this was with working on hamsters with my colleague Uli Munderlo at the University of Minnesota. She's a veterinarian PhD, and she's in the Department of Entomology, and she actually studies Anaplasma and Ehrlichia. And I was working with her, and we had a hamster model. So I was already used to looking for little tiny things. And then I looked to my first foray into trying to image Bartonella in the skin. And I also then put in markers so I could look at the blood vessels and other markers for Bartonella. And what we found initially was I thought I had done everything wrong, because unlike the dogma that says the Bartonella is in the blood vessels, well, it was, but it was also in the dermis. It was like, well, what the heck did I do wrong? Well, as it turns out, nothing. I had access to a lot of very high-end scientific microscopes. And I was actually in Brazil when I did some of this work. And we had skin biopsies stained for Bartonella and put them on this special microscope that can see collagen fibrils in the skin. And that collagen then, and we don't have to stain it. That's a big deal in microscope land when you don't have to stain things. So, but we did have the Bartonella stained, and we found that the Bartonella would just kind of lay right along the collagen fibrils. And then we actually grew some in the lab, Bartonella cultures, and found that it would grow the best on collagen, and it looks like a biofilm. Now, we have a lot more testing, and we are going to be doing that. We'd like to test the hypothesis that Bartonella does indeed form a biofilm on the collagen in the dermis, and that can serve as a reservoir. And as many of you probably know, if you're a Bartonella buddy, you know all about biofilms, and that they can be, not because they are actually inhibiting antibiotics from getting in. Antibiotics can get in, but the Bartonella and the other bacteria and other microbes are actually sleeping. They're metabolically inactive. And for the antibiotics to work, the bacteria and the other microbes have to be dividing. They're not dividing, the antibiotics don't work. So that's another long answer, sorry.
No, that's okay. You're making me think of a lot of questions. So I think the first thing is maybe defining collagen fibrils. I mean, maybe they are what they sound like, but maybe you could.
They are just what they sound like. Yes, they're proteins, thick cables of proteins, and they are what's called the ground substance of the skin. The collagen forms kind of like, and when I say fibrils, that they're kind of like loosely like this.
You said that it's found all over in the skin. Do you find it more in lesional skin than in non-lesional skin?
The Bartonella?
Yeah.
Yeah, it seems that that's the case, but that certainly doesn't mean it isn't elsewhere. When we've compared and we've taken a biopsy of, and when I say lesional, I'll say the ones I have looked at is typically just the Bartonella tracts. And I know I'll just talk a little bit about that. Or we could also call them non-conventional striae distensae. Okay, striae distensae are the typical stretch marks that one might get with, if you're gonna have a baby. Obviously things are stretching, lifting weights, growing really fast. And they're taking steroids, those conditions can cause kind of the formation of these stria, but they tend to be parallel to one another, and they tend to be bilateral. They're on both sides. Now, what we find with the Bartonella tracts, and the reason I've taken to calling them tracts, because when someone says stria distensae, they automatically go to, oh, that means it's caused by such, and such.
Yeah.
And I say, no, I totally disagree with that. I think the typical striae distensae are typically caused from stretching, scar formation from stretching. And I think with Bartonella tracts, now I don't know the mechanism, but it seems as if the collagen is disrupted and scar-like. So it's different. There seems to be more Bartonella found in those lesional tracts than non-lesional, but that certainly doesn't mean that they can't be found elsewhere. And I'll go back to my son because he's given me permission to talk about his history of his disease. Is we've never biopsied a lesion on him, and we've always found Bartonella. Now, this is one of the things we'd really like to study, because what we don't know is we don't know why. We have a lot of ideas as to what could be happening, but none of which I think we can really, what I can say is that they are present and they typically can be found, not always. They typically are found in young people, say teenagers, and I believe that's because they're actively growing and actively laying down new collagen. But do I know that for sure? No, that'd be another hypothesis we would want to test. They do change color sometimes. For example, when one takes antibiotics, you may find that they'll blanch, they'll become more white or more red. And a lot of that may just be a local inflammatory response, could have something to do with the microvascular system. Hemodynamics could change, blood flow could change, inflammatory response in different cells coming into the area. We don't know. So we have a lot of work to do, but I just am so intrigued in my own mind, having seen what I have seen with this kind of microscopy called second harmonic generation. We're able to see the Bartonella right along the fibrils of the collagen. And to me, it may be something as simple as just disruption along that fibril. And that's what causes that, what to us looks like a linear formation. But people will have them on their face, on their arms, on their leg. Just it's only one-sided typically, so it's very different from the conventional striae distensae. But I think an indicator that the physician should test for Bartonella.
So in prep for this interview, I wanted to learn. I read a fair amount about striae distensae. To get a base knowledge of that in order to compare and contrast to the Bartonella tracts. So, I see a lot in the patient groups, I see a lot those aren't striae, those are Bartonella tracts. And it's we don't know that. We don't know. And then also, I think people don't know that the word striae is the medical term for stretch mark.
That's right. Exactly. Yes.
So
yeah.
That's the first thing about stretch marks. And then I read a study that looked at 300 pregnant women and found that younger pregnant females, especially teenagers, were more likely to develop stretch marks in pregnancy than older females who are pregnant. So that was interesting. And then also genetic factors are at play.
Yes, of course. Yeah.
So females that have stretch marks during pregnancy, significantly more likely to have other family members who got stretch marks during pregnancy.
Sure. I didn't know that. That's interesting.
So the changing color, I think people go, oh, well, at the beginning of treatment, they were red and now they're white. And it's well, normal stretch marks do that too. They become red or violaceous, and then they lose pigment and they become indented. And then normal stretch marks often form perpendicular to the stretching.
Correct.
I also read a very interesting article they used 9 young people, so not a huge amount of participants from ages 16 to 22. And they took samples of very early striae distensae from both the middle of the stria, non-involved skin, and then the leading edge about 0.5 centimeters away.
Oh, yeah.
And they found, I thought this was really interesting, that stretch marks follow a well-defined sequence of histopathological changes. And so I'm forgetting to breathe. Breathe, Jake.
It's exciting. I know, it is.
I am riveted. And so first, what they found was on that leading edge where, the stretch mark was probably going to continue to develop. that looked normal to the naked eye, there was actually degranulating mast cells near elastic fibers accompanied by elastolysis, or the death of the elastic tissue. And then the next thing that happened, and it's still not visible to the naked eye, is they found activated macrophages appear. And then the last part is when the stretch marks first become visible, is the collagen bundles show structural alterations. There's macrophages and fibroblasts, and then the mast cells are no longer there really anymore once it becomes visible to the naked eye. So, I mean, that was an interesting study. It sort of made me feel like, oh, well, this seems kind of like an inflammatory process in these young teenagers who
Sure.
are ostensibly healthy. But then my other thought is, well, how many of those might have had Bartonella?
That's a good question. Yeah. Yeah, that's right.
So,
That's absolutely right. And those things you just mentioned are all really, really valuable tools in trying to understand the Bartonella tracks that,
yeah,
you know, that, and again, I only call them that. I call them that based on when I saw the light rail train in front of me, and where the little accordion is, it says, see tracks, think train. Okay, and I thought, well, see tracks, think Bartonella. Okay,
I like that.
yeah. And do we fully understand it? No. The study you just talked about is just really informative and may play a role, and obviously does play a role. And then we're going to throw in, you know, the bacteria as well. And
Yeah.
all of those processes could all just be churned up as well.
Yeah.
So yeah, to me, it's just an indicator that we
You should be looking for
need to
Bartonella.
look for it. Yeah,
Yeah.
right. And there are people that have both, obviously, too.
So I've been sort of stuck on, why can't they be stretch marks? Just so in maybe in someone who is already going to develop stretch marks without Bartonella. I mean, you can't do this. This is just a hypothetical. So
Yeah,
um they were already developed stretch marks, but with the presence of Bartonella, with the damage and maybe inflammation to the collagen, they develop more severe stretch marks. And then maybe in
quite
people,
possible. Yeah.
and then maybe in people who weren't going to develop stretch marks in the absence of Bartonella, maybe they developed mild stretch marks. And so I guess that could be part of it. One other question is, maybe what's the difference between stretching and growing? Like stretching, maybe during pregnancy, it's stretching. And then if you're a teenager growing tall, that's growing. Is there difference in stretching and growing in the skin?
Don't know that. I don't know that. I would say they'd be different, you
Mhm.
know, and it also depends again too on, you know, where the force is exerted on these people as well.
Yeah. So when you said you see it unilaterally, I see a lot of pictures of bilateral. So can you explain more?
Unilateral means you'll just find them here.
Hmm.
That's it.
And you see
Or
that?
just here, you know.
Do you see that? You see that often?
See it often meaning what? Just one side?
Yeah.
Typically, yeah, but I can't say that, you know, like you said, if one is prone to that, would you maybe expect them to be bilateral?
And have you seen them on people's faces? Because I haven't seen any. I don't think I've
I
seen.
have seen pictures of the face involved. Yes, I have.
So.
It's just been like a few
Mhm.
marks there. And you know, like here, this isn't from the skin stretching like this.
I mean, who has
Or
stretch marks on their
yeah,
face?
no, or even in the places where we do find it. And yes, and I think you made a good point that if you are prone to that,
Yeah.
then maybe there is some mixture of the two.
Yeah. Yeah.
And but I don't know that.
When I first got sick, I would come across all the skeptical articles on the internet and stuff, and I came across like this MD who wrote striae due to Bartonella is a stretch, and I'll give him that it's a cute name for the article.
I know
And
this article.
yes, so I mean, I think from a scientific point of view, he makes some good counter arguments that like we should be thinking about, and if we want to prove causality or whatever. And he talks about he talks, I thought this was funny, doing an animal model and seeing if you give these Barton, and then I was like, do animals get stretch marks? And I don't know if this is true, but I did find an article that says that some Rolls Royces use leather from bulls so that they don't have stretch marks, because maybe cows do get stretch marks. I don't know. I just thought that was funny. I don't have a Rolls Royce. I will never have a Rolls
I don't
Royce.
have one either. No, no, just a Toyota, that's all.
If we find them in the things that look like stria more, is it a chicken or an egg phenomenon? And I guess we probably don't know the answer to that yet.
Nope, nope,
Yeah.
we don't know that. We are actually going to hopefully be active. We are going to be actively engaged in trying to parse out what's going on with Bartonella and biofilms. I mean, we have a lot of work to do. We have what looks like visual evidence of biofilms of Bartonella in the collagen of the skin. Let's see, Annette Motor in Germany is working with visualizing Bartonella biofilms in the heart and with endocarditis. And Monica Embers is working with biofilms. Bob and I are working with biofilms. A lot of people are, because I think we're understanding how important they are. And I also work with another guy, Vince Rotello, though we haven't connected since COVID, but he is a biofilm expert, and we'd like to do more work with him as well. I would say we're really in the infancy in understanding it, but I think the medical and research world is definitely at a place where we understand it's very important.
Yeah.
Really important.
When you see the biofilms underneath the microscope, what do you see?
It's a mess. It looks like a big mess.
Okay.
It looks like a big old mess, and I'm not kidding. So the ones that I can, of course, see the best are the ones we grow in a culture dish.
Mm-hmm.
And they literally look like you taken like a old muffin and made crumbles, and there's crumbles all over. But this is at the microscopic level, okay? So, and honestly, you can see them. You can go like go look at a boat, and that line where the water hits the outside of the boat and it's kind of slippery and dirty and slimy. That's a great biofilm. Or just go in here and you got one.
Yeah.
Or in here and you got one. You know? So yeah, and you can't see them, but you see evidence of them.
Yeah. And then are you finding that they are just Bartonella? Or are you finding there are multiple genera, different genes?
Yeah, different microbes.
Different microbes.
We haven't done enough work on that yet.
Okay.
I think we really have to understand what's going on with just one microbe. Then we can look more. Research has shown that the biofilms are multi-species.
Yeah. Well,
so
it makes sense.
It makes sense. Yeah, it makes sense. And I'm sure you've seen some of the great review articles on biofilms in the journal Nature and Science, and some of the journals that have great review articles, and they have very good imaging of what's going on.
Okay.
So,
I haven't seen that, so
I will send it to you.
Okay.
I'll send it to you. That's pretty cool. Yeah.
I don't think I totally understand when stretching's not involved at all, like how it's damaging it, and maybe because we don't know yet, or what's your
We don't know yet.
Okay.
So we don't know yet. But if it is as it looks structurally under the microscope, it looks, instead of being nice waves like this, it's kind of.
I saw the picture. I saw the picture. I was showing it to my mom. I was like, see, this is where like it's lesional tissue, and then the non-lesional tissue I was saying looks like Starry Nights by Van Gogh, because it would look.
Oh yeah, that's great. Yes, yeah. That's perfect. Perfect. I love that. Yeah. I think I'll just have to change the colors to match that idea. Yeah, I'll do that. Yeah, and the other one looks like, it looks like a mess. It's definitely, things are going hither and yon, and that it looks like a typical scar. And when you get scarred, they don't stretch anymore.
With what we know about Bartonella perhaps impeding blood flow, you then might not get as much healing from whatever.
True.
Yeah. And then if
Yep,
it's already.
absolutely. I would agree with that. Yep. So that's one of the things I like about being able to do the work on the big pieces of tissue that we do, relatively big, not big, microscopically big, I might add.
You're
Okay.
taking chunks out of people. You're
Yeah,
just
taking, yeah,
chunks.
little bits, little tiny bits, but it's more so than, so for example, typically after you've taken with anesthesia, after you've removed the biopsy and you take thin slices of it, typically they're like 2 to 5 microns thick.
Mhm.
And then the way that developed is because in the light microscope, you need to be able to get light through the sample to see it.
Mhm.
Okay, now we don't do it that way. So we take a piece like this. So
Yeah.
instead of 2 microns, it might be 100 or 200 microns thick. But until the advent of lasers, and now we go down with the laser, and this is, imagine the laser just goes down just this far, only that far, and
Mhm.
it only gets the signal, the fluorescence from that far down. Then the next one goes this far down. Then this, so it's like a mini, I say it's like a mini CAT scan,
Okay.
like taking a picture of each individual slice of bread, putting it back together. But then what we get is the 3-dimensional morphology and natural pathology of the skin. And that's the beauty of that. So we can see, well, what's happened? We found that the lymphatic vessels are really in a mess. They're very different from, I'll say, healthy skin. The blood vessels at first look appear to me to be more kind of bloated, if I have to say, in the skin.
Mhm.
We have not looked at blood flow function. That would be really important. We're looking at nerves. We found Bartonella is associated with the nerves. Well, and that's really bad because it's going to most likely, it's going to in some way impede or speed up the flow of the pain signals. So that's a big problem.
Mhm.
And so we have so blood vessels, lymphatics, the insulating sheath around nerves, myelin sheath, obviously be somewhat distorted. So we have a lot of things to look at. We've talked about mast cells. We've talked about the white cells that are involved.
Yeah.
And then we're going to throw in some microbes into this foray, plus other immune cells. And so it's a big mix, but we're trying to unravel the mix so that we can maybe understand, well, okay, because we know, for example, in Bartonella, you can get hemangiomas that look like big blood-filled blisters,
Mhm.
especially in Bartonella bacilliformis down in Peru. And you can get these little lesions elsewhere. And it's likely that these angiomas, hemangiomas, are inside your skin as well.
Mhm.
So we know that with Bartonella that you get increased growth of blood vessels and lymphatic vessels. This is really bad, just to kind of segue off into that melanoma paper.
Yeah.
If you have cancer, now that the picture is really unclear. And that goes back to your good question earlier on the chicken and the egg. So if you have Bartonella and you get skin cancer, what happens? What if you have skin cancer and then you get Bartonella? We don't know that, and that's where using animal models like mice would be really important. And what we did with the melanoma paper, we found when we mixed malignant melanoma cells with Bartonella Hensley, we got increased amounts of inflammatory markers and increased amounts of VEGFC, which is something you don't want when you have cancer. That's vascular endothelial growth factor. This is bad, and it's actually one of the leading ways to reduce metastases is to stop blood vessel growth and to take a VEGF inhibitor.
Mhm.
So yeah, I think there's a real complex story there. There's other work being done also on inflammatory breast cancer, and I think that could be very important.
Since we're on the melanoma paper, I mean, first, I don't want to freak people out.
No, 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 no.
No, like Bartonella causes skin cancer. We are so far away from that
Oh, no,
conclusion.
no, don't want to say that out. And maybe it inhibits growth. We don't know that. Or maybe it encourages growth. Maybe it has no effect at all.
Yeah.
But when we looked at 8 patients with metastatic melanoma,
Yeah.
that's working with Paul Reicheter and Ed Breichford and the gang, and 7 of 8 of those people all had Bartonella. Well, it's enough to make you want to sit up and take note when you're talking about a deadly cancer.
Yeah.
There's also a treatment for melanoma. And again, I am not a melanoma expert, so my apologies if I'm not saying everything correctly, but what I do know is that tigecycline, an antibiotic, has been used to treat melanoma before.
Interesting.
And I just saying that there is a whole new side of understanding about the role of the microbiome in general in everything that goes on in our body. A recent paper came out right after our melanoma Bartonella paper came out in Nature, I think. And it was a group, and they looked at, I think, 7 different lesional tissue from 7 different people with melanomas. And they did a really beautiful, exhaustive job on defining and characterizing an abundance of microbes within that. It's going to happen. It's on your skin. It's going to happen.
Yeah.
You know, this is just a little forest of microbes. So it's valuable information no matter what.
Yeah.
But we need, it needs more investigation.
I mean, I think
Don't you hate it? Everything you ask me, I say we don't know, and we have to study it more. But that's actually the truth.
but that
We know a lot.
that's good. And what scientists will say is we don't know. They will say they don't know. Yeah. There's a big gap between the scientific literature and then what yeah. And it blows my mind, because you read the scientific literature and it's like microbiome this, microbiome that, infections this, and then you go to doctors, and I'm not here to just badmouth all doctors.
Oh heavens, no.
They have restraints on them. But yeah, it's just, I mean, Dr. Mosiani says, 17 to 21 years, or what does he say? Like science progresses like one death at a time. Like one's like, yeah.
It takes a long time to get and again, as we work it out, we don't have definitive answers yet. And then the textbooks and then the teaching will change.
Yeah.
It doesn't help the patient today. So we're hoping people like you having, this accessible channel, for patients will be helpful. And I think it's really important to be able to communicate.
Yeah. Getting science to the general public more quickly and more accessible. But
as we have just experienced over the last, year and a half with the pandemic. Getting the science out is really important. And what they did was cutting edge science for sure.
Definitely. I don't like to talk about silver linings of COVID because so many people died, but
Sure.
That doesn't mean that what we learned, it won't be very helpful to like translate over to other illnesses. And then about, looking at the microbiome of malignancies, your paper, said that 16% of human malignancies are linked to an infectious agent. And that's what we know now. What are we going to learn in the years to come?
The guy who discovered that H. pylori was a factor. He took this, he self-inoculated, as did the guy who discovered Bartonella. He also self-inoculated, and he died.
Daniel Carrion, yes, and he died.
Yeah.
But that was, in 1890, whatever. Hopefully doctors don't have to do that anymore, and scientists don't have to.
Well, yes, they do actually, but
they still do.
I can't say they self-inoculate, but
you haven't inoculated yourself with Bartonella?
No. But
I wouldn't recommend it.
No, not going to do it. Not going to do it. No.
About the Bartonella in the nerves. So when you take the punch biopsy and you're looking at it with the laser, microscope. Is that similar to the type of punch biopsy that you would get for maybe small fiber neuropathy?
Exactly the same.
Yeah.
And we've looked at that. We're looking at that.
Yeah.
We're looking at small fiber neuropathy.
I think clinically doctors would say they see an association, but we'll
Yes.
need the research to definitively say that, I
Yep.
guess.
Yep. And also, I have done work on that before. And we had a mouse model, and we did find that this was a mouse model for MS. And when looking at the nerves, the peripheral nerves in the epidermis and just in below the epidermis, because it's hard to do that in mice, we did find an overabundance of nerves, and they had more pain. They were more hyperalgesic
Yeah.
than, so I think we have to think of not only neuropathy caused from too few nerves, but also damaged nerves and also too many nerves, all of the above.
Mm-hmm.
And we did some work on cancer pain that showed a similar phenomenon. More pain nerves
More pain
in the
nerve.
tumors. Yeah.
Wow, that's very interesting. So there were 2 mouse studies that I read. One where you inoculated, well, some mice with saline and then some with Bartonella, and they had like more,
Oh, okay. Yeah.
so they had more like hyperalgesia. So you like, I don't know,
Mhm.
like maybe pricked them in the paw and they pull their paw away if it hurt.
Yeah. So what it's fishing line. It's not like a pokey thing. It's fishing line.
Yeah.
They're called von Frey hairs. And
Uh-huh.
they do this on people too. I've
Yeah.
had it done. And they just take fishing lines of different diameter. So, if you have a fishing line that's just 1 finger thick, it's bendable. But if it's 3 fingers thick, then it takes more force. Okay.
Yeah.
So a fishing line on the bottom of the paw. So what we do is we have a little elevated, it's so high tech. It's just some wire, a net, a screen over some 2x4s,
Yeah.
and not even 2x4s. Anyway, we put the mouse on there, and we put like a glass, a dessert dish or something over them so they can't move. And then we just wait till they get comfortable. Okay. And you know that because then they just lay down.
Yeah.
Okay. And then we poke their little foots with fishing wire, and we don't poke them hard. We just say, what width or what diameter of this von Frey hair will cause them to just withdraw their paw. So
Yeah.
it's called paw withdrawal frequency.
Yeah.
And so what we find is that for the mice that have hyperalgesia, that means more pain, more sensitivity to a non-painful stimuli. Okay. And so that means that the mice that have hyperalgesia, they're more sensitive. You just do the skinny wire, the skinny fishing line, and they go, oh, I don't even like that. And if you have a sore, you don't want anybody to touch that.
No.
And
I don't
so
want anyone to touch me when I'm healthy. Just don't
that's
touch
true. So anyway, so it's a standardized way to test for, are your nerves really sensitive or not?
You did a study with mice that had sickle cell disease. You gave some of them Bartonella, and then those mice were more sensitive, probably using the same method, right?
Right. And that would be with my colleagues in Brazil that we did that, and with another colleague of mine who is not at the University of Minnesota anymore. We also found that in sickle cell mice, their lymphatic vessels are really abnormal. There's a lot going on, and as you can imagine, in sickle cell disease.
Yeah.
Where the blood vessels are really rigid and form that sickle shape. And that's why they get stuck and cause lots of bruising and damage in the joints.
I think I read in one of your papers that sickle cell disease is the #1 hereditary hematological disease in the world. So it's very prevalent. 10% of like people of Afro-Brazilian descent.
It's a big problem. And
yeah, huge problem.
One of my colleagues on that paper is now running the blood center down in Brazil.
Yeah.
And for sickle cell. And
Yeah.
it's a big problem. It's a huge problem
for morbidity and mortality.
Right. And then we're finding that, and honestly, this kind of just took off because my friend Kalpna was doing sickle cell and I was doing Bartonella, and we said, hey, what if we do this? And because it could really happen. It definitely could happen.
Yeah.
We also looked at people that had liver transplants, because when you get a liver transplant, your liver is not checked for pathogens, your blood's not checked for pathogens. I'll say vector-borne pathogens, nor are you. So it's both the donor, the recipient, and the blood, no checking. And my colleague in Brazil did find a young girl who passed away because she was given an infected organ. We do know things like Babesia is found in the blood bank supply at the Red Cross.
Yeah.
That was discovered here by my colleague at the Red Cross.
Yeah.
And my friends in Brazil also did a study on blood bank blood down in Brazil in Campinas.
Yeah.
It's a really huge city, and found, I think, I can't remember the percentage, but it had Bartonella.
Yeah.
That was viable.
Yeah.
And that's in the blood bank, for crying out loud.
Yeah.
And we are not testing. Oh,
I know.
and can one get funding for that? No one can't, because I've tried.
You would think that that would get you funding because it's very mainstream, and it's not hard to understand. It's not controversial. It's like you just don't want your blood to have bad crap in it. So
That's exactly right. Yep.
Yeah.
Totally agree with you. So we have a few studies that have been done.
Yeah.
One, again, by the Red Cross. So if you have Babesia, you don't give blood, they won't take your blood, and you shouldn't. If you've ever had a tick-borne disease or even a hint of one, don't give your blood. Do not give your blood. Some people can be infected and asymptomatic, but the next person you give it to, and I think you're right, you made the point about, how long it takes for what we are doing in the research world to filter down into medical school.
Yeah.
And it takes a while. And it's scary that it does.
Yeah. So it shouldn't. I get why it might take a long time, because if you're going to change guidelines for 1 million doctors in this country, for millions of patients in this country, you better be pretty sure about what you're changing guidelines on.
Yeah.
At the same time, it doesn't need to take 20 years.
That's exact. I agree with you.
Yeah.
I agree. And I would think, being in this internet age, things could move quicker. That at least we've learned through COVID, we can overcome what in the past has seemed like insurmountable bureaucratic hurdles to make something happen. It can be done. So it can be done. And that's a good lesson to be learned, I think.
Yeah.
I mean, obviously the need was huge.
Yeah.
Yeah. And I think there are a lot of good people funding it, and it's too bad there isn't more. There is more. There's going to be more now with LymeX, which will be the new Lyme initiative from the NIH and the Cohen Foundation. So that will be good.
And then the last thing about sickle cell was the paper where you found you tested a little over 100 patients and 9% were found to be bacteremic with Bartonella. I know what Bartonella is like without sickle cell, and it can be really painful. Yeah,
of course, it's horrible, and a lot of that work was done with Paulo in Brazil.
Yeah,
and because they have a large population there.
Yeah,
that he works with, and Paulo is actually infectious disease and dermatology both. And so, and he's involved in research, and he spent a year in my lab, and it was just great. It was so great just to have that cross fertilization, of ideas and experiences and knowledge and that we could bring to the table. Of course, what happens to a lot of people with sickle cell is they get blood infusions.
Yeah.
And we've just identified that there's a real need to improve our blood supply.
I've never given blood because I think you have to weigh 110 pounds, and then I'm short, and
Darn, I could. Okay. Yeah. Okay.
But now and there were brief moments where I did weigh over 110 pounds and I felt guilty about not giving blood. And now that I know that I probably had Bartonellosis the whole time, now I don't feel guilty anymore. So go me.
See, you are just smarter than you thought.
So one thing about the stria, I see in the research, there's a lot of connecting it to neuropsychiatric symptoms. And what I've been thinking about that is because, not necessarily because, or these marks, the Bartonella tracts, not necessarily because they're more prevalent in those with neuropsychiatric symptoms, but if you just have neuropsychiatric symptoms and you don't have any physical like pain or GI or whatever, and you have these marks, then that's like even another clue that you don't just have a neuropsychiatric disease. You should be looking for an infectious agent.
Yes.
So is that why you're tying these marks to neuropsychiatric specifically?
Well, yes, and I actually say a lot of this came from Rosalie Greenberg, who has actually she is a child psychologist and a good friend. And she said, what's going on? I'm seeing these stripes, these streaks on these kids. And then, so it was kind of, I mean, I know there's other people than just her, but I think it's also for the observant person looking and saying, is there a correlation here? And if there's a correlation, and you've probably read the few, especially the really fairly recent paper about the young man who, he was in a bad way as far as neurological symptoms. And he had the tracks on him, and they did find indeed he was very much positive for Bartonella, and antibiotic treatment turned him around.
Yeah.
And yes, we'd love that for that to happen with everyone.
Yeah.
And, and this big, to me, it kind of begs the issue, well, what other, and this is just total pie in the sky thinking, but, if we have these microbes that are difficult, that grow really slow, that are difficult to detect, and because our diagnostics have not been, and at nobody's fault. No, there's no fault. It just technologically we weren't able to detect them. And who else is infected with some different microbes of some kind? And and I don't want to in any way say anybody that's got an issue should go get tested for Bartonella. I'm not saying that at all. I'm just saying this is something else to consider for the physician who is actually treating some of these people, and for the patients too, because I know what it's like I say, 2 years and nothing. And, for my son, and literally, like I said, 50 doctors, and no one had a clue. And then it was Bartonella. And he had a lot of neuropsychological issues as well.
And he's doing a lot better now.
He is great. He is, I will say, happily, he is married. They have a dog and 2 cats and some fish. And he's working with pre-K kids at an inner city school and loving it. And he wants to be a kindergarten teacher. I'd say that's pretty darn good.
I would say so.
And he's a wonderful son besides. He's good to his mama. So that's good.
That's what they should be. That's
right. Yes.
That's what I'm gonna find one day. One thing I didn't ask is about, seeing it in the lymphatics. I've read about it being, in the endothelial cells. And so that would impede blood flow. If Bartonella is in the lymphatic system, what might that do to the body? What symptoms might that cause?
That is a gall darn good question. Now, is that just the normal highway and byway for these microbes to go when they've been gobbled up by, different immune cells? Or does that impede just normal lymphatic drainage and immune response in general? Again, sorry, nothing. I have nothing to, because that, again, is not a trivial thing to know. But I think we can be aware of it if someone's having unusual swelling, or if you have, maybe you stand up and then you fall down because your fluid balance is off. There's a lot of things. Everything works in concert. And I have really nice images of, lymphatics and blood vessels like this, and they work very differently, but they're obviously play a very important role.
And they're all, for lack of a better term, like interwoven next to each
Yes,
other.
that's right. They are interwoven. We only look for one thing at a time, and it doesn't seem so packed. And the reason I say that just means there's a lot of interactions we have to consider.
Totally.
It's not going to be a silver bullet. As I say, it's going to be a golden glove that's going to take care of us, which is why we need to do lots of supportive care.
Definitely.
Whatever that means to whoever whatever it means.
One question I have is these marks, even though they're technically a cosmetic issue, I'm not going to act like people aren't distressed by them emotionally.
Oh, no.
And so
absolutely.
I was wondering if, just knowing what you know about the skin, if you knew of anything that might either prevent, mitigate, or help the appearance of them after they've already developed. And so I know for stretch marks, you can use like a tretinoin cream, not during pregnancy. And then you can also get like laser. I don't know if you have thoughts on those, or if you have additional thoughts.
I really don't. And that is something where you really want to work specifically with a cosmetic dermatologist, someone that does laser therapy.
Yeah.
And would know about that, or someone that does a lot of work with scars, and someone in your area that does that. And
I mean, it's expensive, so
it's horribly expensive. Yeah, it is. I know, but just to say, they aren't giving those lasers away, and they don't last. They don't. You have to have them replaced every so often.
Yeah.
So I don't really feel comfortable even addressing that at all.
Okay. I was just hypothesizing, if you're going to do that laser treatment, maybe do it after you've healed.
Oh, I think that's reasonable. Yep, I would have to agree with you on that. Yep.
Yeah, after the inflammation's calmed down, so then you can have a better healing process, because it is scar-like tissue. You'd want the best. So they with laser, they kind of damage the tissue a little bit so that you can heal back up. And so
that's right. Yep.
You'd want good healing to happen. So
that's right.
If you're gonna laser those marks.
Sure.
Can you give us any specifics on what you're working now with the microscope, what maybe we could expect the next thing to come out? And then, or no?
What's hot? Yeah, no, that's good. I mean, we really want to do more work on the melanoma, and try and understand that a little bit better. So, we would like to actually do a kind of a retrospective and maybe a prospective study on just biopsies from people. Is it different depending on the classification of your lesion? Do you have the Bartonella? Do you have Borrelia? Well, I would say a big thing we're really working on is multiplexing, trying to look at for more than one microbe at a time. That's definitely one. So also looking for oncogenic and inflammatory markers, like what's important here. And we can use different cell lines. So we can use melanoma cells that are somewhat benign. We can use them that are highly metastatic, and we can use different kinds of Bartonella. We can mix in Borrelia with that. We have actually looked at some of the melanoma biopsies had both Borrelia and Bartonella. We didn't publish on the Borrelia. Or is just this, if we look at a lot of people, so we're going to look at controls as well, are we going to find the same incidence when we look in the skin and not just at the blood? So far, we found it in brain, Bartonella, that is.
Yeah.
Brain. It's been found in eyes and in skin. And Dr. Breitschwitz found Bartonella in dog saliva. I think cat saliva too, but don't quote me on that one.
I just did a video on saliva. I haven't released it yet, but I just did a research video on saliva.
Oh, you did? Oh, good. Okay. So then we're going down the body here.
Yeah.
And we've also found it in heart and published on that.
Yeah.
And these are all things we've published on. We found it in ears, dog ears. I'll mix human and animals together. And
Are
Then,
animals, so it's
yeah, yeah.
fine.
Lymph nodes, spleen, liver, appendix. Haven't looked at the colon yet, but we're going to be doing that in bone as well. So as Dr. Mosiani will say, if someone says, well, it's all in your head, well, the answer is yes, it's in your head, but it's all over too. It's not just in your head.
And it can
So
cause
yeah,
almost any symptom or no symptom at all.
that's right. And Dr. Embers just published an elegant paper showing Borrelia in the brain as well.
Yes, I
Not surprised. Yep, we are looking at more of that. And the other thing we're doing is we're doing what's called deep sequencing. So instead of like PCR, which can be very sensitive, but you only can pick up what you're testing for. Working with Dr. Bemis to really look, and there are others too. Dr. Ehrlich is doing a lot of that as well, and other people. I don't mean to negate anyone. We're really looking at the whole biome, the microbiome, and how this can be different. And of course, we'd like to know, why is one person sensitive and one not? That's obviously the key. And what's the key to getting better? How do you get better?
Yeah.
You know, how do some people, like at COVID, have a and some people don't make it at all?
Yeah.
You know, there's a lot of stuff we don't understand.
Yes, there is a lot of stuff we don't understand. You know, when you read in the scientific literature about Bartonella, VEGF comes up a lot. And then when patients go to test for VEGF, my VEGF wasn't elevated. A lot of people say that theirs wasn't elevated. So the question is, I mean, maybe you don't know the answer, and that's fine. But is it because
that's what I always say. Okay. Yeah.
no, maybe this is more of a clinician type question, but so anyway, is it because the VEGF might be very localized and then the tests aren't, testing it like in the cell? I mean, I don't know how VEGF works. Is it because the tests aren't sensitive enough? Or is it because it only does VEGF in certain patients under certain conditions?
You've answered your own question. And no, where we have tested it is in the Petri dish and in the samples. And we're looking at localized areas. So it may be very much localized. So that's very much true.
So then your blood level wouldn't necessarily be.
May not. And as we all know, tests are not what we would always like them to be.
No, and labs don't have to publish anything on any of their testing methods ever in this country, which this is the axe I grind every day.
No, I'm with you on that. Yeah. And at least if it comes out of a research lab, we have to tell you what we did. It would be nice to know. Because
we, as a patient, paying thousands of dollars for these tests, I mean,
and hopes. It's the hope too. It's the hope that you're going to find something.
And then people latch on to a positive when it's like, well, maybe that's a false positive. They go, oh, this is a good lab because it gave me a positive. A good lab doesn't give you what you want. A good lab does good science.
Yes. We could be on the same team.
We're here.
Yeah, we are. My thing I keep driving home is, out of all the controversies that we have in this vector-borne disease world, good science and good communication is going to get us the answers. And we have to be asking all the right questions, and we have to ask the hard questions. Say for some that you asked today, and I could say, I don't know, but I'm going to say, I don't know. Before I pretend to know something. I'm never going to say that. I can say, here's what I've seen when I've looked at this in the Petri dish. Does that have anything to do with what's going on inside? A little, maybe. I hope, but not always. There's a huge problem. We use mouse models because they're very, very good, but you can't directly correlate what you're doing in the mouse is going to happen in the human. And I think this is particularly a problem, and we have a lot of really good researchers working on this, is that a lot of the mouse models, the rodents, are really good at clearing Bartonella.
I've heard that from
Yes. And this is a,
Dr. Elam, yeah, Amanda.
yeah, that's, yeah, I was gonna say Monica, yep. And yeah, and Amanda, yeah. And this is a problem.
Yeah.
And so, but there are ways to manipulate those, the mouse genetics, so that you can hopefully get around some of that.
And
And
chronic, and have them have a chronic infection instead of just clearing.
yes, yeah, that they can't. Yeah, and is it, and in the, but is it even going to be the same? And just as long as we don't try to overinterpret, we'll be okay.
Yeah, you know, people go, oh, they found Bartonella in my blood with a microscope, and so I'm just like, okay, this organism is so tiny, like dude with his microscope in his basement is probably not seeing what he thinks he's seeing. So can you explain to our audience and me, you know, maybe the finer points of that?
Sure, I think I can.
Being a microscope expert.
Person, yes. So honestly, with the microscope, if you're just doing what's called bright field, okay, where it's not fluorescence, and you know fluorescence is, oh, like the fish that glow in the dark, right?
Yeah.
Okay, that's actually really high-tech science from about 10, 20 years ago. And now you can buy these fish, and I am buying some in my aquarium this weekend, actually. Anyway, so, but we do have naturally occurring things that fluoresce and phosphoresce. So our eyes and the microscopes we have are physically, by the laws of physics, you cannot see those microbes. They're too tiny. You can see a spirochete like Borrelia, but you're not going to see Bartonella.
But you can also see artifacts of spirochetes, and they're not actually, you can see pseudospirochetes, but that's a whole nother.
Yeah, okay. So, okay. Anyway, yeah, I'm just saying size. I'm just going size.
Yeah, size. Okay.
Okay. No, so the way that we visualize them, we can do electron microscopy, totally different ball game. And that's, nobody has those in their basement. If you do have something that you're looking at, you may be looking at a consequence. It's possible, you know, and that's reasonable. For us to see them, we put a fluorescent tag on. So if you ever are looking at these things where you can look at the holiday lights, and then you put on those funny glasses and it makes them look really big, you know, when you look at a light. So we're essentially, that's what if you consider my hand, the microbe is here. And what we're looking at is we're looking at the light bulb effect.
Yeah.
And even so, it's very difficult.
Yeah.
Because it's very difficult to say, is this really Bartonella or is this schmutz? This is a problem. It's not, and I know it's funny, but it's really not funny to me, because we have to be very careful. So the way we get around that is we have a lot of different controls that we do to do that. So, and that's how we get around it. And we look at it in different ways. We look at it, just now we're looking at RNA FISH. So what the heck does that mean, Marna? So, you know, you're in your body have DNA, and that's kind of like carries the code, the code, and then you have the RNA, which is the machinery. It's like it's you may have the tar to lay down on the road, but it's not going to get there unless you've got the machinery to put it down there. Yeah. So that's essentially what RNA is. And that's really good, because often RNA will mean that organism is active and alive. It's growing, because one of the comments is say, oh, you're only looking at something that's dead. And my comment was, you got that much dead stuff in you, you're in a jam, no matter what, I don't care. But nonetheless, so we look at RNA, and then we take a little piece of RNA that we know this is Borrelia or Bartonella or Babesia or whatever, you know, and then we make the copy, and then the copy goes on, and then there are different ways you can do this, and then you put this fluorescent bulb on the end of it, essentially, and then you say, oh, there it is, I can see the light, right? And believe me, many microscope companies, Nikon, Zeiss, Leica, have spent billions of dollars on research to figure this out. It's not trivial, but I mean, at least we know we can do it now. And now with RNA FISH, the FISH means fluorescence in situ hybridization. So in situ means in the tissue, and hybridization means just hooking them together. So we're hooking together an RNA probe and the fluorescent light bulb. That's all that means.
Okay.
And then I do that with antibody staining as well. I have an antibody to Bartonella, and this antibody, and there's different ways, I'll just say, to amplify the signal. You can do that as well.
Okay.
Okay, and I'm not going to go into all of that, but there are ways to, you know, if I only have this signal, but if I put these amplifiers on it, that only goes here, then I've got, imagine a whole bunch of hands around. So then you, and again, that's one of the issues we have, and we have to be very careful about not, especially with Bartonella, it's tricksy, is not confusing it with something else, because all the tissue we look at, all the blood we look at has what's called endogenous fluorescence, meaning some of the stuff just lights up anyway. And
what are those things? Do you have some examples?
some of those things, well, let's take the best example I have is collagen, okay? But only under certain conditions. So I can look at collagen, and because it lights up using, okay, I'll do this. It's a multiphoton laser microscope with second harmonic generation imaging. Oh, what a mouthful. Anyway.
I read the papers and I'm like, I'm never going to get this.
Well, I barely get it. So anyway, but what it is it allows some linear molecules like collagen, actin and myosin. It's actually the myosin. There are some others that will actually have a signal because of the way it bounces the light around inside. Okay. Bartonella has endogenous fluorescence. So you can see it in the green channel. And think of the rainbow of light, the spectrum of light. Well, we take advantage of that and we isolate different parts of that rainbow. And that allows us to look at different colors. Hemoglobin is a horrible one. NADH, other energy producing molecules, lysosomes in the heart, just about killed me when we did that heart paper because they endogenously fluoresce. We took advantage of the endogenous, that means when we hit it with a red light or a green light, in this case, we hit it with a laser at 488. And then we looked at what came off of that. And we said, oh, look, there's a big bright signal from Bartonella at this wavelength. And this one is, anyway, you do a subtraction thing. We can do that. We can use this to our advantage, and we just have to be aware of it when we're looking. And then because I said we can isolate different probes, different reporters to use different parts of the spectrum, different parts of the rainbow of light. We're going to look at red Bartonella, and I'll call it the green channel. And I'm going to look at a nuclear probe just for DNA in the far red. And then I might do blood vessels in the red channel. And so we can do those all separately, and then we can mix them all back together.
Yes. What I'm getting from this is you need a high level of expertise and experience, and you probably need these three letters after your name, PhD.
Yeah. Well,
probably.
I have to tell you a little story. When I was in high school, I took one science class, just one. And that was biology, general biology, okay? And it was the seniors and the sophomore, everybody was together. I turned the page in the book, and I think I was 17, and I saw a picture of a drawing of a mammalian cell. And they had done it in gray and red. Okay, fine. And I said, oh, that's really cool, right? This is like 1,000 years ago, approximately. And then I flipped the page, and there was a green cell, and it was a plant cell. And I said to myself, right then and there, I said, that's what I'm going to study. So yeah, I did. That's what I did. So I'll just tell you, I got my bachelor's degree in botany and my PhD in plant physiology. In between my bachelor's and my PhD, I worked in a hemoglobin chemistry lab. Okay, then my boss said, oh, okay, so then I got my PhD and then I needed a job, but I had started doing work on confocal microscopy. It was brand new then. And I got a job in the Department of Dermatology. And which was, now you say, Marna, why are you a plant physiologist? But I'd had 6 years training in a hemoglobin lab, so I knew stuff. It's just a general biology degree with plants, focused on plants. But anyway, so then I get to derm. It was the best thing because I didn't have any blinders on. I didn't have any like, oh, it's supposed to be this.
Mm-hmm.
I had none of that because I didn't know anything.
Mm-hmm.
So I had to learn everything. And that was actually most absolutely to my advantage when I did the work on pain in bone tumors with a mouse model. I said to my colleague, the neuroscientist, I said, oh, George, look at all the nerves and the tumors. And he laughs at me. We're both at the microscope and he says, Marna, nerve, but tumors don't have nerves. And I said, but George, there they are. There they are. They're in the tumor. And then that led to a giant project. And we made tumors that fluoresced red. And we had markers for the different, for nerves and blood vessels and pain nerves. And yeah, and guess what? Those people that in the mice, not the people, the mice that had were hyperalgesic, frequent paw withdrawal. Anyway, they had a lot more nerves with something called CGRP, which now people take for migraines. It's involved with pain and nerves. Those mice with the hyperalgesia that were hyperalgesic with the tumors had a lot more pain nerves in their tumors than those that did not.
Yeah.
So, and I will tell everyone, how do you know if you don't look? You don't know if you don't look.
You don't know. You don't know.
And if you look, you might be very surprised and actually, you have a new way to look at. That's what I do. I look at stuff.
Yeah.
And I love doing it. I love it. So that's good.
It is good. I think those are all my questions, but I did want to give you the opportunity if there's anything you wanted to talk about, Bartonella, skin, microscopes, whatever. Is there anything you wish I asked you or you want to add?
Write to your congressperson and tell them to give more money to vector-borne disease research. That's what I'll say.
That's good with a professional, nice tone.
Yes. So I think it would behoove all of us to listen to the best science that's available, the scientists, physicians to do the best, not only the best science, but the best medicine, stay current and try to stay open to new ideas. I think there's a lot to learn and encourage your congressperson to allocate funds for studying vector-borne diseases and the ramifications that we all, unfortunately, some of us have to suffer with.
Yeah, patients are mostly who watch my channel. I didn't know if you had any words of encouragement or advice for those who are going through that right now.
I would say keep looking, keep trying.
Yeah.
And don't give up.
Yeah.
And if you don't find help with person A, Dr. B, whatever, then go someplace else. Keep looking. You have to keep looking till you find something. And I would say trust your gut. I had so many people that told me, it's all in his head. He just doesn't want to go to school. And I honestly flat out said, no, wrong. That is not the right answer. He loves school. That's when I hear that, my blood boils. I'll tell you, I just get so angry. I'm sure you do too.
Yeah.
Sure. So,
yeah.
Okay.
I think that's really good advice. And I would say the same thing. Hi, everyone. Editing Jake here. This is where our interview ends because Marna and I just started chatting about a whole bunch of other stuff. So we didn't do a formal goodbye, but don't worry, I thanked her. Please enjoy this clip of my mom doing a mock interview with me pretending to be Marna. It's very, very convincing of an acting job, if I do say so myself. And you found B. Hensley via PCR from 2 patients with melanoma, and from a 3rd patient, Bartonella was amplified from the blood PCR.
How do
So
you know every freaking thing about me?
it's all on the internet, Marna, for people to read. It's called a Google Scholar.
No.
Did you- when you were when you were publishing these things, Marna, did you not know that they were going to be public?
I had no idea. Nobody told me.
So you don't know that published, usually it's for people to read. That's what published means.
Well, I would have made it better if I knew people were going to read it.
How were you going to make it better?
I don't know. I wasn't going to lie.
Did you know that lying in published scientific did you know that lying in published scientific literature is extremely unethical, Marna, and you can be reprimanded for doing so?
Oh, bad me.
No, like you can be barred from publishing, and it might be hard to ever publish again.
Oh, big whoop.
You could go to jail.
They're going to catch me first, right?
Are you really fast? Okay, well, thank you so much for being with us. This was enlightening. And
You're very welcome.
yes,
And good luck to you.
Yeah, Godspeed to you too.